Chapter 13, Section 3, DNA Replication. Learning objectives. Explain the role of DNA polymerase in copying DNA. Compare DNA replication in prokaryote cells and in eukaryote cells. Recall the general DNA structure: antiparallel strands, sugar phosphate backbone, nitrogenous bases A equals T, C equals G, and it's a double helix structure. Before a cell divides, it duplicates its DNA in a copying process called replication, during the S phase of the cell cycle. During replication, two strands of each DNA molecule separate. Two new complementary strands are then synthesized following the rules of base pairing. Each strand of the double helix of DNA serves as a template or model for the new DNA strand. DNA replication is semi-conservative, which means that each DNA molecule resulting from replication has one of the two original strands and one new strand. During the S phase, the two strands of the double helix separate, allowing two replication forks to form. As each new strand is synthesized, new bases are added, following the rules of base pairing. If the base on the original strand is adenine, then thymine is added to the newly forming strand. Likewise, guanine is also paired with cytosine. For example, a new strand that has the base sequence of TACGTT produces a strand with complementary base sequences of ATGCAA. The result is two DNA molecules identical to each other and the original molecule. DNA replication is carried out during the S phase of the cell cycle by a series of enzymes. These enzymes first unzip a molecule of DNA by breaking the hydrogen bonds between base pairs and unwinding the two strands of the molecule. Each strand then serves as a template for the attachment of complementary bases. The principal enzyme involved in DNA replication is DNA polymerase. DNA polymerase joins nucleotides to synthesize a new complementary strand of DNA. Besides producing the sugar phosphate bonds that join nucleotides, DNA polymerase also proofreads each new strand so that each molecule is a near-perfect copy of the original. The tips of eukaryote chromosomes are known as telomeres. Because the ends of DNA molecule are difficult to replicate cells using a special enzyme, they use one called telomerase to do the job. This enzyme adds short repeated DNA sequences to telomeres as the chromosomes are replicated. In rapidly dividing cells, telomerase helps to prevent genes near the ends of chromosomes from being damaged or lost during replication. Telomerase is often switched off in adult cells. In cancer cells, however, the enzyme may be activated, enabling these cells to grow and proliferate rapidly. In most prokaryotes, DNA replication begins when regulatory proteins bind to a single starting point on the chromosome. These proteins then trigger the beginning of the S phase, and DNA replication begins. Replication in most prokaryote cells starts from a single point and proceeds in two directions until the entire chromosome is copied. Often, the two chromosomes produced by replication are attached to different points inside the cell membrane and are separated when the cell splits to form two new cells. Compared to prokaryotes, eukaryote chromosomes are generally much larger and more complex. In eukaryote cells, replication may begin at dozens or hundreds of places on the DNA molecule. Then it proceeds in both directions until each chromosome is completely copied. Several proteins check DNA for chemical damage or base pair mismatches prior to replication. However, the system is not perfect. Damaged regions of DNA are sometimes replicated, resulting in changes to the DNA base sequence. These are called mutations. The changes may alter genes, which can cause serious consequences. The two copies of DNA remain closely associated until the cell enters prophase. At that point, the chromosomes condense and the two chromatids in each chromosome become clearly visible. They separate from each other in anaphase of cell division, producing two cells, each with a complete set of genes coded in DNA.